Information Science University, and this is my dissertation work, which is late breaking, still kind of in process. So this is, start off stock photo to tell you about diversity. Obviously, a bunch of people on a street are different. But how are they different? Different in a bunch of different modalities that we can kind of think of as being independent. So maybe genetics and life history, we can assume have no real influence on each other, maybe, or we can treat them as different independent modes when we're trying to model. So that's on a whole organism level, but this is also true on the cellular level. So for individual cell, it's genetics and epigenetics, and maybe if it's a microenvironment or how it's being treated are all kinds of orthogonal dimensions on which to think about how it influences the response. So one area in which this is, I think, going to become increasingly important is precision medicine, where we're trying to figure out which compounds or which drugs are going to work best for a given patient. We can't just hand them all the drugs and see which ones work. So we need another way to try to get at this combinatorial difference between these different modes of response. One way of doing that is to, instead of treating the patient, we treat cell lines with different drugs at different doses in these large-scale cell line panels, and then kind of measure the response of the cell line in the presence of the drug at that dose, and try to make connections there between the cell line genomics and the drug. So the idea here is we want to do drug recommendation based on some of this data, and also discover relationships given the data. But what do we mean by response? The studies up to date have taken the data from these types of panels where you have increasing dosage of the drug along the x-axis, some measurement of cell growth along the y-axis. You normalize that to a 0-1 range normally, squash it down into some curve or fit some curve to it and extract maybe one value from that, and then make some cutoff about whether the cell line's responsive to the drug based on that singular value extracted from all the data that you had. So it's a huge amount of data compression, and it's really mathematically impossible to fully summarize a curve with one number. So can we do better, and can we move towards this kind of multimodal modeling? So my idea is let's try to predict the whole dose-response curve and ask questions about it after that. This is a type of multitask learning. So for every cell line drug combination, I'm going to be trying to predict a number of values, the response at each dose. My inputs are gene expression, mutation, copy number, whatever kind of omic data I can get on the cell lines. And for the drugs, I'm also using predictors that can be just targets of which targets we know genes the drugs target, or some structural elements, whether they contain certain side chains and stuff like that. And the output is going to be this predicted growth inhibition at each dose. So this is kind of a natural way to think of the data, where I'm holding everything together in one object, this three-dimensional matrix, or a tensor. It's kind of the simplest way of thinking of tensors. It's not a complicated physics definition. It's just a cube of numbers, right? For every cell line, drug, and dose, I have one response value. And in this case, as I move to increasing doses, I see more blue. This is kind of indicating the cells are growing less at higher doses for all these different drugs. So I'm going to be trying to predict this as my response. How do we make sense of an object like this? There's kind of some old ideas of taking tensors and breaking them apart into some core interaction, smaller tensor that compresses the data a lot, and matrices for each of the dimensions to explain what's going on there as well. So my model is based around this idea of tensor factorization. And this is a general view of the model that I'm calling baffled, to stand for a Bayesian tensor factorization linked to external data. So the central tensor response is here. I have predictors for each of the modes that are multiplied by some projection matrix that's going to reduce the dimensionality of the inputs drastically. And I want it to be sparse in some ways so that I select which of these predictors are useful in predicting response. So I squash down each of these input data into some latent representation, and latent representations for each dose combine to form the response tensor. So if I'm thinking drugs and cell lines, this would be one cell line multiplied by one latent factor in order to give me a latent representation for that first factor for the cell line. And I build that up across the multiplication. Then once I've done that for each of the input modes, I take the outer product of these latent factors. So in the latent matrix, each row is one cell line or one drug, and each column is one latent factor. I do an outer product of this, which gives me the full response tensor, the number of cell lines by the number of drugs, by the number of doses in this case. And the core tensor gives me some weight on an interaction between those latent factors. So the outer product here times that weight gives me one full response tensor. I do that again for another latent factor. I get a different weight on it and a different response tensor and add those all together to build up the full response. So this is the model. And in specific for this problem, I don't have any input data for doses, so we can just leave that off and just have a latent representation for the information in doses here. So once we train this, what can we do with it? If I have if I have missing data in my response tensor here, just factoring it into the latent representations and multiplying those back together fills in all the missing data. So this is a kind of imputation task where I'm predicting the response for a cell line drug combination where I've seen responses for that cell line and that drug, but not that particular combination, which is nice, but it's not necessarily using the input data, and it doesn't allow us to predict for a new cell line or a new drug. That's why we need this linked to external data part of it and the extra structure over here. So with that extra structure, if I have a new cell line, I can just multiply it through the projection matrix, latent matrix. So for a new test cell line, I get a set of responses for all the training drugs at each of the doses. So this would be a set of dose response curves for that new cell line with every drug in the training set. Similarly for drugs, I can get a set of dose response curves for a new drug for every cell line in the training set. Or for a cell line drug combination, a totally new, hasn't been seen in the training data, I can get one dose response curve as an extra piece of this tensor. Okay, so how do I actually train this? There's a large number of values depending on how much input data I'm going to use and the size of the latent dimensions that I want for each of the modes here. There's a lot of values to learn. It's basically too big to try to do some Monte Carlo sampling method. We want to have probability distributions on each of those values in order to have some idea about variability and make the model more stable. So I'm using, that's what I want to learn, projection, latent matrices, and the core tensor. And I'm using a variational approximation algorithm to do that. So this is, it's been mentioned, I think, in a couple previous talks today. But it's a way of getting at this really complicated joint distribution across all these variables by approximating that distribution with a simpler factorable distribution and then minimizing the difference between that factor distribution and the true posterior distribution. It proceeds by starting off with a random initialization of the values that I don't know and then updating all the values in the model mostly sequentially given expectations of the other values in the model. And this allows for training to be done in a reasonable amount of time. So the data sets that I'm looking at, starting with a smaller data set from the DREAM7 challenge, this is all breast cancer cell lines treated with different drugs. And a large data set that was released a little over a year ago from the Cancer Target Discovery and Development effort, which is the biggest to date of this kind of data, around 900 cell lines, 550 drugs, and more doses. This is a pan-cancer cell line, so it's much more difficult to kind of learn the latent factors across the different types of cancer. And just for perspective, this is kind of a tiny corner of the big data set. So here are some results in comparison to simple metrics that I've done so far for this DREAM7 data. So the DREAM data, I haven't actually tested on the original held out cell line yet. There's 17 held out cell lines that I don't feel like quite confident to test on those yet. So this is for a 10-fold cross-validation and how I perform in comparison to different elastic net models. So ridge regression, elastic net with different values, and lasso. And then this mean indicator here is for a new cell line, I would predict the response for all the cell lines in the training data set, the average of the mean for all the cell lines in the training data set for that new test cell line. Or for a new drug, I would predict the mean for all the training drugs for that new drug. So it's a simple baseline, but my algorithm so far is able to beat those baselines in these different tasks. So the warm prediction task of kind of filling in missing values in the tensor, the test cell lines, a kind of cold prediction task where I'm predicting for a all new cell line and I get a full suite of curves for all the drugs in the training set. Test drugs where I get curves for a new drug for all the training set cell lines, and a new cell line drug combination, getting one curve. And I was just showing Pearson correlations there. So for those same, if I want to drill down a little bit more, in these types of problems, people are generally interested in how are you doing on individual drugs. Some drugs are going to be easier to predict than others. So if I look at the prediction on a per drug basis for a new test cell line, and this is kind of a head-to-head comparison between that mean prediction, which the mean, if you remember from the previous slide, the mean prediction was about on par with all the elastic net models. So here the Spearman, or the Pearson correlation for the mean prediction is on the x-axis, and for my model on the y-axis. So if I'm above the diagonal, then my model is predicting for that drug better for a new cell line. This is kind of the mirror for a new drug. It's harder to predict, so each dot in the original here represents one drug. Each dot here represents one cell line. So for a new drug, it's harder to predict, but my model's still doing slightly better than the mean prediction. And for a new combination, it's kind of in between the previous two. I'm able to predict a new drug for a new combination by drug, each dot here being a drug. The majority of them are predicted better by my model. And just to show you what some of the curves look like, just looking at correlations as response, it's difficult to decide here what the best response measure is. No one's tried to predict these response curves before, so it's a new problem. So just looking at some of the curves can be interesting. So this is just an example where the curve is predicted well. Here predicted well, it's at maybe missing one dose. So this could affect the GI50 if you were trying to predict that. Here the curve's kind of showing the right behavior, but maybe the growth rate is wrong. So you notice the x-axis here are different, and it's because I'm modeling kind of the raw data, not normalizing it to a standard growth 0 to 1 growth range. And here I get kind of the right behavior, but it needs to be maybe scaled somehow. Also, we can look at how the sparsity and what predictors are being used for these predictions. So this is just an example of some of the matrices, these projection matrices, and what they look like. The way the package is set up now, you can decide whether you want to share the variance across the rows for predictors and either select a predictor for all the latent factors or not, which is what it was in this model. And it's kind of pulling out certain pieces of data to be useful. For cell lines, it tends to pick up the subtype indicators first, as they're much more useful in explaining a response. And then if I look at the latent matrix for doses, it kind of picks up some of the typical behavior, something like a principal component analysis, where the first latent factor is pulling out this general gradient slope. As I increase dose, I see a lower growth. Moving on to the bigger data set, this is still very preliminary results, but I'm able to, first of all, the elastic net models do much better than the mean here because they are able to learn the growth rate, I think. And my model does on par with those. But the amount of data compression here is really large. So this is a model where my core is only 8 by 8 by 8 tensor. So I'm really shrinking down this huge amount of responses to a small kind of latent representation of what's going on. And I'm confident that I'll be able to improve on these types of prediction responses in the future with larger models. But it is difficult to run this algorithm in enough time. So with an 8x8x4 model on this large data set, it takes about a day and a half, 2 days to run on a 12-core machine. So with that, I just want to kind of summarize and think about in general. So the algorithm that I've developed, I'm applying it to this task of predicting these drug response curves, but I think that the uses for it could be much more broad. We can think of many different types of modes of variation, anything where we can measure some multi-dimensional response and have predictors for those. So now it's implemented only in 3 dimensions, but I think we need to start thinking in higher dimensions in order to really make sense of the kind of data that's starting to be produced by these types of studies. So I'd like to thank my committee, especially my advisor Kamal Samas, who's here, and the other members of my department, other fellows, my wife and friends and family, and my funding source from the National Library of Medicine. This is my email. If you're interested in this, email me. I have a working model up on GitHub now that you could download and play with. And that there's a vignette that walks you through generating a toy data set and learning it. And it's depending on how you set up the toy data set, the size of it, it can learn the predictors very well, perfectly, and the responses almost perfectly. I've got a poster tonight. And I'll ask for questions, and while I'm answering questions, I'm just going to play a video of the model learning on the toy data set so that you can see it first kind of fits the responses, and then after 10 or 12 iterations, you start to see sparsity developing in these projection matrices. This is the core matrix, or slices of the core tensor, which I didn't mention also have distributions on there to encourage sparsity so that I pare down the interactions. Not all the interactions between latent factors are actually used to make the responses. So you'll also see sparsity kind of developing in this as it learns the model. So, thanks.